Now let us understand the characteristic features of the enzymes or enzyme catalysis. Now remember those catalytic processes or catalysis in which the enzymes are the main responsible catalysts. They are having following characteristics. First is most highly efficient. Remember, they are having very high degree of efficiency. Even one enzyme molecule can catalyze, can transform 1 million molecules of the reactants. So this is how you can imagine how strong or how efficient they are. Second, which makes them very separate from the normal usual catalyst, and very important one, are their highly specific nature. You cannot replace one enzyme with the other one. Like if urease is breaking down the urea molecules into ammonia, if you are trying to use the apapsin or some other enzyme for this purpose, it won't be possible. So they are highly specific, they cannot be substituted by any other enzyme, so they are almost indispensable at a particular stage or for the particular reaction. So they are highly specific, they work only on certain specific reactants. The reason mainly is because they all are shape selective catalysts. All enzymes are having active sites or specific pore sizes on which only those reactants are absorbed. That is why they are highly specific to a particular reaction. Then remember, they always work under certain optimum temperature. We know our human's body temperature usually is 37 degrees Celsius. So you need to understand they only work from between temperature 25 to 37 degrees Celsius or 298 to 310 Kelvin. That's their optimum temperature. They cannot work below this, below 25, but they cannot work efficiently above 310 Kelvin or 37 degrees Celsius. And that is why it is advised that we should not be exposed to a high fever. Always the doctors try to bring down your body temperature, because these enzymes their enzymatic activities get stopped or disturbed the moment the body temperature is above 37. Then there is another important thing. They always work under optimum pH. The pH maintained by them is always, the human body maintains a pH of from 5 to 7. So they always work between the pH of 5 to 7. And remember, whenever the pH is changed due to the taking of higher intake of salts or due to some other reasons, these enzymes get killed. They doesn't work. Their enzymatic activity gets stopped. So in order to make them work efficiently, the pH should be between 5 to 7. Then next, activators and coenzymes. Remember these words, coenzyme. Coenzymes are certain compounds, non-proteinous compounds, which gets attached, or they are always found to be attached with the enzymes, and they are the one which further enhances or increases the enzymatic activity, or they increases the enzyme's catalytic process. So such non-proteinous molecules, compounds or substances or chemical species which are attached always to the enzymes and increases their enzymatic activity or increases their catalysis are called coenzymes. Most important of them we can understand are vitamins. Vitamins are coenzymes. They are always increasing those enzymatic activities. Therefore, their immediate intake, if they've stopped, it doesn't affect much. But yes, if regularly they are absent in our regular diet, in balanced diet, definitely those specific enzymatic activities gets affected. Then let us understand activators. Activators are the minerals which we take, like sodium, cobalt, copper, zinc, etc. All these ions, basically they further increases the activity of the catalyst. They activate the catalyst by attaching themselves with these enzymes. So remember, these ions again are very important in our diet, basically because they are binding themselves with the enzymes and increasing their enzymatic activity. Then the last and the most important, the inhibitors and poisons. Remember, inhibitors, those which reduces their activity of the enzymes. Or poisons are the chemicals which binds to those active sites of the enzymes, changes the shape of their active sites, or doesn't allow the reactions to be absorbed on them, and therefore they either slow down their enzymatic activity or they completely stops them. Such chemical substances are called inhibitors or poisons. So they should be avoided to increase or to have the maximum utilization of the enzymatic activity. So these are the main important characteristic features of the catalyst or enzyme catalysis. Now let us understand the mechanism of enzyme catalysis.